Hi folks, and welcome to another edition of Zog Science. Today we're going to be talking about the discovery of DNA's structure and the scientists that contributed to that discovery. So the discovery of DNA structure really begins in earnest early in the 20th century. This basically took about the first half of the 20th century, so the early 1900s, and it involved several different scientists. The first group was T.H. Morgan's group, Thomas Hunt Morgan's group, and they were the first group to show that genes are located on chromosomes, and the composition of chromosomes was known to include both DNA and protein, and so those became the candidates for genetic material. And the key factor in determining the genetic material was choosing the right experiments, and it was solved by using studies of both bacteria and viruses. So the first one was the idea that DNA can transform bacteria, and this was from research by Frederick Griffith in the 1920s. And he worked with two strains of bacteria, one that was pathogenic, which means that it caused disease, and one that was harmless. So what he did was that he mixed heat-killed remains of the pathogenic strain, so basically he heated them up to kill them. He mixed that with living cells of the harmless strain, and when he did that, some of the living cells became pathogenic. And this phenomenon is called transformation, which is basically horizontal transfer of genetic information. So rather than transferring genetic information from one generation to the next, you're transferring that within the same generation. And it is also sometimes defined as the change in the genotype and phenotype of an organism due to assimilating or taking in outside DNA. So again, the way that the experiment worked, he took living cells that were pathogenic, which are the S strain, and gave them to the mice, and the mice died. He then took living R cells, which are the non-pathogenic ones, the harmless ones, injected it into the mice, and the mice lives. Then he took heat-killed S cells and injected them into the mice, and when he did that, the mouse was able to live. And so then finally he injected a mixture of the heat-killed S cells, the pathogenic cells, with the living harmless R cells, and when he did that, that caused the mouse to die, and he was able to take out and culture living S cells from the mouse. So essentially the DNA from the L cells was being incorporated by the living R cells and turning them into S cells, and that is the process of transformation. So later work by Avery and others identified what the actual transforming substance was as DNA, but other biologists still remain skeptical, mainly because little was known about DNA, and they thought proteins were better candidates for the genetic material. The reason why they thought that was that they knew that DNA only had 4 bases, whereas proteins were made up of 20 amino acids. So they thought that the proteins were much better candidates for passing on massive amounts of information because there were so many different amino acids. So the evidence that actually proved that DNA was the genetic material came from studies of viruses that are infecting bacteria. And those types of viruses are called bacteriophages, and they're used in a lot of molecular research. And so a virus is either DNA or RNA enclosed by a protective protein coat, and viruses must infect cells and take over their metabolic machinery in order to reproduce. And the way that they do that is by injecting DNA. Well, at the time they didn't know that, and so they conducted an experiment in order to figure it out.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T2 phage, which consists of DNA inside a protein coat. The reproductive cycle of the T2 phage begins when the tail fibers of the phage stick to the receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes are used to manufacture phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures, or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the reproductive cycle of bacteriophage T2.
So in 1952, Alfred Hershey and Martha Chase showed that DNA is the genetic material of the phage known as T2. So that kind of diagram you just saw, they didn't know exactly how that functioned at the time. And so to determine this, they designed an experiment showing that DNA was what was going into the bacteria and the protein was not. So the way they did this was that they took radioactive isotopes of sulfur and phosphorus, and they made the protein coats of the phages with the radioactive sulfur, and they made other phages that were radioactive DNA using the phosphorus. The reason why you can do that is that sulfur is only found in proteins and phosphorus is only found in DNA, at least when you compare them to each other. So what they did was that they allowed those phages to infect cells. And then they did a centrifuge where they spun down the results. And what they found was that only the radioactivity was found inside the cells, while the of the DNA, and the radioactivity from the proteins was found outside the cells. And so because of that, they were able to conclude that the viruses, the T2s, are injecting the DNA into the cells, which is what is then allowing them to take them over, whereas the proteins are staying on the outside, and therefore they were able to conclude that proteins are not the genetic material and DNA is the genetic material. So at this point, it was known that DNA is a polymer of nucleotides, each consisting of a nitrogenous base, a sugar, and a phosphate group. And in 1950, Chargaff reported that DNA composition varies from one species to the next, and the evidence of diversity made DNA a more credible candidate for the genetic material. So the structure of DNA is you have each nucleotide, which consists of a deoxyribose sugar, a nitrogenous base, and the phosphate. And we have an alternating phosphate sugar backbone. You also notice the directionality of DNA. The directionality comes from the 5' carbon. So the 5' carbon, you start over here on the right, you count 1, 2, 3, 4, 5. So the 5' carbon is open on the 5' end, and then the 3' carbon is open on the other end. And you'll notice that it is the 3' carbon is where the phosphate will bind here on the next one. Okay. And there's just a little bit closer up so you can see a little bit more clearly. Here's the 3', here is the 5' end. So the two findings became known as Chargaff's rules, which is that the base composition of DNA varies between species, and also that in any species, the percentage of A and T bases are equal, and the percentage of C and G bases are equal. Now, at the time, they didn't really understand exactly what that meant, but what we know now today is that A pairs with T and G pairs with C, and that's the reason why those percentages are equal. So Watson and Crick were the first people to actually determine the structure of the DNA. However, they didn't really do any experiments themselves. They just built on the work of other people. So Wilkins and Franklin were using a technique called X-ray crystallography to study the molecular structure of DNA. And so Franklin was actually able to produce a picture of the DNA molecule. And the picture is what showed that DNA was made up of two strands in a double helix. So here is a picture of that. You can see how we have the helical structure here from the X-ray crystallography. And the other thing that it helped them to determine was the width of the helix itself, which was able to also confirm the fact that A should pair with T and G should pair with C.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar-phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar-phosphate backbones.
So here we can see the structure a little bit more clearly here. We've got one strand on one side and one strand on the other side. Notice that these strands are moving in opposite or facing in opposite directions. The 3' end of one strand is the opposite of the 3' end on the other strand. So DNA is an anti-parallel molecule, which is something we've looked at before when we were looking at beta-pleated sheets. The reason why A pairs with T and G pairs with C is due to hydrogen bonding. A and T form 2 hydrogen bonds with each other, while G and C form 3 hydrogen bonds together. All right, thanks for watching, and I'll see you next time.